The last part of this chapter that we're going to talk about is a continuation of protein synthesis, mainly the process of translation. So we talked about transcription was going from from DNA to mRNA. Now we're going to talk about the process of going from mRNA to proteins. So what does the word translation usually mean? If you were thinking that it means to change from one language to another language, you would be correct. So if we talk about translating something from English into Spanish, it means that we're taking whatever the language is in English and basically just keeping the meaning but changing the letters and the words to Spanish. So in the process of translation for protein synthesis, it's decoding the mRNA into a new language of proteins. We're moving from the language of nucleotides, which was our DNA and RNA, to the language of amino acids, which is the language of proteins. So if we take a look at this picture, you will notice that I have a nucleus, and the type of RNA that's leaving my nucleus is mRNA. That mRNA is going to the ribosome, where it will go through the ribosome, and transfer RNA will bring the amino acids to the ribosome to make the new protein. So the process of translation. First, my mRNA travels from the nucleus to the ribosome in the cytoplasm or attaches to the rough endoplasmic reticulum. Remember, ribosomes are either free, meaning floating in the cytoplasm, or attached, meaning they're attached to the rough endoplasmic reticulum, giving it the rough appearance. The mRNA is read 3 bases, or 1 codon at a time. The first codon of any mRNA strand is AUG. So if we take a look at this, transcription was going from my DNA strand to an RNA strand. That RNA strand then moves to my ribosome, where it is translated into my protein or polypeptide, 1 codon at a time, meaning every 3 bases gives me 1 amino acid. Those amino acids are then put together to give me 1 protein, or 1 sentence. The third step is the tRNA, with an anticodon that matches each codon, delivers an amino acid to the ribosome and is then released to attach to another amino acid. We'll take a look at this in just a minute. An enzyme helps to form the peptide bonds, or the strong bonds, between the amino acids to form a protein. And the tRNA continues to deliver amino acids to the ribosome until a stop codon is reached. In other words, a 3-base sequence of mRNA that tells it to stop translating the message. So if you take a look here, we have a tRNA with its anticodon UAC, which is being released from my mRNA because it has already dropped off its amino acid. So if you look at this part, my tRNA is coming in, its anticodon is attaching to an mRNA codon, and when it does that, it will move through the ribosome and attach its amino acid. As a result, I start to get this long polypeptide chain, or also known as a protein. The newly formed proteins can do one of the following. It can then be used by the cell. It can be stored by the cell, in other words, for instance, stored in a vacuole or a lysosome, or it can be shipped to another cell. For example, sometimes hormones are shipped out to new cells to signal messages. If you take a look at this circle chart, this is how we are able to determine what amino acid is going to be added to my strand. So, the inner lighter circle is my first base. My a little bit darker yellow middle circle is my second base, and the very small letters in the outermost circle is my third base. After I know what sequence of three bases I have, I'm able to determine what the amino acid is. So, for example, A, U, G is going to code for methionine, or a start amino acid, something that's going to begin the translation process. If I take a look at, for instance, U, C, G, it's going to code for serine. The last type you might have is UAA, which would be a stop. It's not actually an amino acid, it's just a signal to tell it to stop translating. So we're going to show the process of translation using the mRNA molecule provided. So here is my mRNA molecule, and my first step is to determine which amino acids are going to be coded for. So if I look at my circle, AUG codes for methionine, ACG codes for threonine, GCU codes for alanine, and UAG codes for stop. I don't need the circle anymore in order to determine what the tRNA anticodon is. I just need to write the complementary strand. So A pairs with what when I'm doing RNA? If you were thinking U, you would be correct. So my first tRNA anticodon would be UAC. My second one would be UGC. My third one, CGA, and AUC. These are based on my complementary base pairing rules. The central dogma of biology states that the sequence of DNA determines the sequence of RNA, which in turn determines the sequence of amino acids. Therefore, the number and sequence of amino acids determines the size and shape of a protein. The size and shape of the protein then determines the function of the protein. So if you think about it, DNA ultimately determines the function of a protein.